Welcome students to this module of e-learning. I am Dr. Maria A. D'Souza, Assistant Professor, St. Xavier's College, will present to you this module today. To the students of First Year Botany, Semester 1, Course Code BOC 101, Course Title Biodiversity 1, Microbes, Algae, Fungi, and Bryophytes. The title of the unit, students, Unit 3, Fungi, Module name, Agaricus, Classification and Morphology. The outline of the module, systematic position, morphological characteristics and structure of Agaricus. The learning outcomes. Once the student completes this module, the student will be able to give the systematic position of Agaricus, describe the morphological features and structure of Agaricus. So students, today you will be studying about Agaricus, its classification and morphology. Agaricus, students, is commonly called as mushrooms, and it will come up mostly during the rainy seasons or where there is sufficient moisture. So students, let us begin today's class on Agaricus, its classification and morphology. This is a picture of Agaricus, commonly called as mushrooms. The systematic position of Agaricus belongs to kingdom Fungi, division Eumycota because these are the true fungi, subdivision Basidiomycotina because the sexual spore which is produced in basidiomycete is called as a basidiospore. Class Hymenomycetes because the basidia and the basidiospores are initially covered. The hymeneal layer is initially covered with a membrane, but on maturity, it exposes. So, it belongs to Hymenomycetes. Subclass Holobasidiommycetidae because the basidium which is formed on the hymeneal layer is a septate. Order Agaricus, Agaricales, which include all the gilled mushrooms. Family Agaricaceae. These are the mushrooms which are found on dead and decaying organic matter. And the genus Agaricus. Now, Agaricus, students, you can see the picture. You can see the picture of Agaricus. Now, Agaricus is commonly called as mushrooms. Most of the species of Agaricus are saprophytic in nature. They are called as meadow mushrooms because they grow on open, well-manured grassy areas, on damp, dead and decaying substances such as dung, humus, rotten wood, twigs, trunks. Some of the edible species of mushrooms like Agaricus campestris, Agaricus bisporus. Agaricus, the body, can be studied under two heads. One is the vegetative mycelium, which is found living within the soil or the humus where the Agaricus or the mushroom grows. The second part is the basidiocarp or the fruiting body or the proper mushroom as we call it. It's aerial. Above the soil it comes up and it is formed as a result of sexual reproduction. The vegetative mycelium, students, the first part, can be differentiated into primary mycelium, secondary mycelium, and tertiary mycelium. The primary mycelium is monokaryotic in nature. That means it has a single nucleus, short-lived, and it arises from the germination of a basidiospore. Now, if the basidio is of a plus strain, you will have a primary mycelium of a plus strain. And if it is the basidiospore is of a negative strain, then the mycelium arising from the negative strain basidiospore will be a negative strain primary mycelium. The primary mycelium is haploid in nature, which consists of hyphae which are uninucleate, septate, hyaline, with short cells having oil globules, vacuoles, and protoplasm. The secondary mycelium arises from the fusion of the plus strain primary mycelium and the negative strain primary mycelium, and it forms a dikaryon. Now, dikaryon is formed because of the process of dikaryotization, wherein it undergoes successive division and forms the dikaryotic mycelium. The dikaryotic mycelium is binucleate. There is no fusion of the nuclei, and this mycelium have hyphae which are long, perennial, branched, septate, hyaline, with short cells and forms an extensive branching network. This mycelium also has a typical septum called as the doliposeptum, which is found mostly in the basidiomycetes, in which its opening is guarded on both sides with a parenthosome. This mycelium, the secondary mycelium, is most abundant, perennial, and will form the tertiary mycelium, and this tertiary mycelium will give rise to mushrooms year after year. This is how your doliposeptum looks like. This is the hyphal wall, students. This is the septum which has a dolipo, and it is surrounded by the parenthosome. The tertiary mycelium is the mycelium which is formed from the secondary mycelium, and this mycelium produces the basidiocarp or the proper mushroom. The hyphae interlace or twist to form microscopic, thick, white, tangled, woolly hyphal cords called as rhizomorphs, which will bear the fruiting bodies or the basidia, which will bear the basidiospores. The dikaryotic mycelium shows centrifugal growth. The fructifications develop at the hyphal tip of a circular colony, forming a fairy ring. This is how, students, you have the basidiospore here. One will be of a plus strain, one will be of a negative strain. Now, this mycelium is primary mycelium, student. It is short-lived, you can see. And here, it results when it fuses in a formation of a dikaryon. Okay. Now, this dikaryon multiplies, the successive divisions takes place by means of clamp connections. Okay. And it results into the secondary mycelium. The secondary mycelium, students, is very, very long-lived. Now, this mycelium will give rise to the tertiary, and tertiary will give rise to the mushrooms. Basidiocarp, or the fructification, is the aerial portion of the fungus, and it is commonly called as mushrooms. It's an umbrella-shaped, conspicuous, well-defined structure consisting of the stipe and pileus. The stipe is a stalk-like portion. Okay, and it is broader, swollen at the base, and is centrally attached to the pileus, 6 to 9 centimeters in height. Whereas the pileus is the expanded portion of the mushroom. And in young condition, when it is in a button shape, a covering or a thin membrane surrounds that button mushroom, and it is called as a velum. Now, as the button mushroom increases in size or grows into a proper mushroom, the velum breaks open and the remnants of the velum surround the stipe, and that is called as the annulus. So, this, student, is the annulus. This is the pileus. This is the stipe. Okay, this is the button mushroom stage. Now, on the lower surface, on the undersurface of the pileus, you have several thin vertical plate-like structures. I mean these. Okay, and these are initially pink in color, students, and on maturity, when the spores are formed, they will change its color to brown or dark brown. Okay, and this place, students, is responsible for the formation of the spores or the fruiting or the reproductive structures called as the basidiospores. Now, the negative geotropism of the stipe, the diageotropism of the peleus, and the positive geotropic nature of the gills helps the basidiocarp to stand erect. Now, if you see the structure of a lamellae or the gill, it has three parts: hymenium, trama, subhymenium. The hymenium is the fertile region, students. Okay, and it's with club-shaped binucleate cells. Okay, and it's lying on either side of the lamella. All the cells of the hymenial layer are not fertile. There are some shorter cells which are sterile and they are called as paraphyses. Whereas there are some cells which are much elongated, they protrude out and they are called as basidia. And they are fertile and they will produce the basidiospores. The trama is the central sterile region and it consists of loosely arranged interwoven mass of plectencamatous tissue. It has long slender hyphae. These hyphae are longitudinally with multinucleate elongated cells that extend from the hyphae of the peleus with the long axis of the hyphae lie parallel to the hymenium. The subhymenium, students, is situated on both sides of the trama and is composed of hyphae at right angles to those of trama. The cells of this trama at the subhymenium is isodiametric, binucleate, and is formed from the lateral branches from the hyphae coming out from the trama region. So if you see, students, this is the lamellae or the gills which are found on the undersurface of the basidiocarp. If you take a vertical section of the lamellae, you will see three regions. First one, the trama region, which is made up of plectencamatous tissue, longitudinal it lies, and hymenial layer isodiametric cells. Okay, and the hymenial layer, this one, which is made up of fertile cells. Okay, now this layer, hymenium, has fertile cells called as basidium and the shorter sterile cells called as paraphyses. Now, on the basidium, which is aseptate, students, will bear short peg-like structures, okay? Short pointed structures, which are called as pterygmata. And on top of this, you will have the basidiospore. The basidiospore will be having only one nuclei. Now, there are 4 basidiospores arising on one basidium. 2 will be of a plus strain and 2 will be of a negative strain. Students, we have studied today the systematic position of Agaricus. We have also learned what is the structure of Agaricus, how it looks. How do we look at it? It has 2 portions, the basidiocarp and the stipe. The stipe is a stalk-like portion and the basidiocarp is the expanded portion which will bear gills on its undersurface or lamellae. If we take the section of the gills, we have seen 3 regions, the trama, subhymenium, hymenial layer. The hymenial layer will bear pterygmata on which we will have the basidiospores coming out. And the basidiospores, when the favorable conditions arise, will be liberated and they have the capability of forming the primary, secondary and tertiary mycelium. These are the references, students, the offline references, the online references. Thank you, students, for a patient hearing.